Hi, Sarah here, and today I'm going to talk to you about the life cycle of HIV, the human immunodeficiency virus. So here's just a quick schematic of the virus itself. It consists of two strands of single-stranded RNA in an envelope which has these surface antigens, and the two major ones we want to think about are GP120 and GP41 because they're important in helping the virus to bind to the host cell. The host cell of interest in HIV is a T helper cell, or a CD4 cell, and the surface of this cell has got particular receptors that enable the virus to bind. We've got the CD4 receptor and the CCR5 co-receptor. So what happens is the virus comes to the surface of the cell, and there's a complex that forms between the GP120 and GP41 on the virus and the CD4 receptor and the co-receptor. So this is binding, and once this has happened, it enables the viral envelope to fuse with the cell surface membrane, and this is called fusion. And then the single-stranded RNA is able to enter the cell. And what happens now is the opposite of transcription, whereby DNA is converted into RNA, and the enzyme reverse transcriptase does what it says on the tin, and it converts the single-stranded RNA into DNA. So we now have a DNA copy of the virus's RNA, and this can then be incorporated into the host's own genome. And this allows the virus to effectively utilise the host cell's machinery to copy its DNA, or its RNA. So the technical term for this is integration. The viral DNA is integrated into the host's genome. And from here, it can undergo transcription. So once again, we've got an RNA copy of the viral genes, and after it's been transcribed, this RNA can then be used for translation, just like in protein synthesis. So here's the endoplasmic reticulum covered in ribosomes, and I'll just show a ribosome here to show that this is the site of translation of the viral RNA to create proteins, the viral proteins. And then these can all be packaged together nicely, assembled to form another virus. So here we have the RNA, the viral envelope proteins, and that can then come off of the membrane, and this process is called budding. And then the virus matures until we have a mature virion, and that virion can then infect another cell,